Hello, my name is August, and I worked with Dr. Deak on my project, which was about the role of about the role of a protein called synaptobrevin 2 in cognitive impairment. So, cognitive impairment is caused by diseases like Alzheimer's, and when it comes to Alzheimer's, most cases are late onset, which means that they occur mostly in older people. The question here is, why is that the case? Why does it occur in mostly older people? Since levels of the synaptopathy 2 protein decrease with age, the hypothesis for my project was that low levels of this protein impairs synaptic transmission, which causes neuronal connections called synapses to become more vulnerable to Alzheimer's. And since older people have less of this protein, they will become more vulnerable to Alzheimer's. And that's why it mostly occurs in older people. So to test this, we looked at the rate of synaptic vesicular release via exocytosis of neurons with Alzheimer's and either normal or low levels of synaptobrevin 2. So before I get to an experiment itself and what I did, so for the people who don't really know neuro, like the juniors who are watching, I'm going to quickly give like a crash course on some neuro so you guys can better understand what I'm going to be talking about. Although there are a lot more people here who know what I'm talking about than I expected. So, but disclaimer, I'm going to glance over a lot of details, but I'll tell you enough to get the gist of how it works. So neurons are cells in the brain which communicate with each other through a process called synaptic transmission. Synaptic transmission takes place between two neurons and starts with an electrical signal called an action potential, travels down a neuron and reaches the end of it. And at this point, the membrane of the neuron becomes depolarized and eventually exocytosis occurs. During exocytosis, synaptic vesicles, which are basically sacs that contain neurotransmitters, fuses with the plasma membrane and releases the neurotransmitters. So during exocytosis, synaptic vesicles basically just let out whatever it's got inside. After it does that, endocytosis occurs, and the synaptic vesicle membrane is recovered and refilled with neurotransmitter to be reused for exocytosis. So basically, after exocytosis, where the synaptic vesicles fuse with the plasma membrane, the synaptic vesicles eventually come back as part of the membrane. So the neurotransmitters that have been released will cross the gap between the two neurons, called the synapse, and bind to the receptors of the other neuron. And now one of two things can happen. The result is dependent on the neurotransmitter released, but the neuron will either create an action potential and the signal continues being passed on to the next neuron, or nothing will happen. And this is basically the process of synaptic transmission. So finally, onto my experiment itself. In order to find out whether or not synaptobrevin 2 makes neurons more vulnerable to Alzheimer's, we looked at the rate of synaptic vesicular release via exocytosis in neurons with Alzheimer's. So to do this, we used FM dyes, which are fluorescent dyes. This means that they emit light after absorbing light of a specific wavelength. So here's the idea behind it. So we had neurons take up FM dyes into their synaptic vesicles through endocytosis. And because the FM dye is fluorescent, if exocytosis occurs, the synaptic vesicles will release the FM dyes they have inside and consequently lose some fluorescence. If we keep track of this loss of fluorescence, we can find the rate of exocytosis of the neuron. So what I did for the experiment is as follows. I'm not going to go into all the complete details because explaining all the prep would be too much. So this is a general idea of what I did. So first, I had either the neuron with the normal or lower levels of synaptobrevin 2 on a little piece of glass that's part of this larger plate. It's in a little divot on it. So, and there are magnets that secure the glass in place so it doesn't shift all over the place. Then I put the plate under a microscope and look for a region that has a lot of synapses. So after I add the FM dye, there will be a lot of bright spots and data to look at. On the screen, I've put up images of what the neurons look like under the microscope. Of course, the image isn't exactly right because I didn't take any pictures myself. I found these all online and I thought these were close enough and gives a decent idea of what it looks like. In reality, the background was all green and the neurons were green as well, which made it sort of difficult to find a good region. The neurons don't show up as clearly as well, but I think you get the idea. So at this point, I turn off all the lights in the room because leaving them on will mess up with the FM dye and it'll show up too bright when I look at the fluorescence. Then, so now I finally add the FM dye to a depolarizing solution of high potassium and then put it onto the little thing of glass to trigger the exocytosis of the neurons. The depolarizing solution causes the synaptic vesicles to fuse with the plasma membrane, exposing the synaptic vesicles to the FM dye. The synaptic vesicles are then endocytosed with the FM dye inside. So on the right, you can see images of how that works. So in A, you have, before the adding the FM dye. In B, you have the addition of the FM dye and the depolarizing solution. In C, that's where you have the exocytosis, and that's where you have the synaptic vesicle picking up some of the FM dye on the outside. And D is the endocytosis, and where the synaptic vesicle goes back in with some of the FM dye. So next, I wash away all the FM dye and the depolarizing solution, so exocytosis stops. And basically, the only FM dye left is in the synaptic vesicles. You can see that on the right, how there's no more FM dye on the outside, but there's only FM dye on the inside the synaptic vesicles themselves. So while I'm doing this, I basically turn on a vacuum that sucks up excess solution so it doesn't spill everywhere. On my little image I drew over there, the vacuum tube is the little curved line thing sticking up next to the neuron. So this setup is actually hooked up to a computer, which has a program that emits a light attached to the microscope to repeatedly blink, and that lights up the neurons. So this light is of a specific wavelength that the FM dye absorbs, so that the dye emits light that the computer can pick up. The light emitted is shown on the computer screen as those bright white dots over there. So now on the computer screen, we can tell where the synaptic vesicles with the FM dye are. And since we now know where those synaptic vesicles are, we know that the synapses are basically in the same spot. So at this point, I check to make sure that I've got a good region with lots of synapses so I can get good data. I then mark the spots where I want the computer to measure fluorescence, and then I run a program that basically repeatedly causes exocytosis. So you can see what the sample looks like after I finish running the program on the computer screen right there. The FM dye has been released because of exocytosis, and the synaptic vesicles have lost fluorescence as a result. If I compare it to the previous one, you can see that there's a visible difference in the fluorescence. So the computer keeps track of this change in fluorescence over time and graphs it. The graph on the computer screen is what it looks like, and it can be used to find the rate and amount of exocytosis. So now that I have my raw data, I have to do some analysis on it. And by that, I basically mean I have the computer do it for me via Excel workbook macros that hurt my brain. They get very confusing. And so after analyzing all my data, this is sort of what it looks like. And this is the important graph right here. On this graph, the gray line represents the Alzheimer's positive neurons with lower levels of synaptobrevin, while the orange lines represent neurons that are Alzheimer's positive and have normal amounts of synaptobrevin. So this graph basically shows the distribution of release rate as a fraction of the total release fluorescence at 15 seconds. And because you can see that the gray line, which represents the lower levels of synaptobrevin, is more to the left, basically, and the peak is there, it basically shows that the rate of exocytosis is lower than the neurons that have normal levels of synaptobrevin. So from this, it can basically be concluded, the conclusion here is that lower levels of synaptobrevin make synapses slower and weaker, and that's why lower levels of synaptobrevin lead to Alzheimer's. Thank you.
All right, thank you, August. So does anyone have any questions? So I have a question. So August, if if you were to continue this project, what do you think the next experiment is that you would do?
So if I were to continue,
Mhm.
let's see. I think what I would do is I would test it with different neurons of like with different genes
Mhm.
to see how maybe that could affect it.
What
And maybe
other
it's dependent on that.
yeah, what other genes do you think would be interesting to look at?
Uh, I'm not entirely sure on that one, but
Okay.
yeah.